I'm going to talk about species tree estimation. So this is a phylogeny, which is an evolutionary tree with different species in it. And you're probably used to seeing this. It's saying human and chimp have a more recent common ancestor than they do to the other organisms there. This looks like it's a biology talk. This is actually, in some sense, a math talk. So this is a math talk because what you're going to do is look at the mathematical models of evolution and think about how to infer evolution from data. So the connection to data is that you can't do this without data. And also, it's really interesting statistical estimation problems. So dealing with genomic data and trying to infer a species tree essentially on all life is what I'm going to be talking about. But this is just to give you an example. So how would you try to get a species tree from using genomic data, like DNA sequences from different genomes? You would basically collect data, which is here DNA sequences, from the different organisms. You would do some pre-processing, and so they get into a nice thing called a multiple sequence alignment. And then you would use that multiple sequence alignment to try to infer the tree. So you get a DNA sequence here for orangutan, gorilla, chimp, and human, and try to infer the evolutionary process. Statistical estimation, okay? So we talk about this in the context of genome-scale data, and then you get something called phylogenomics, phylogeny estimation using genomic scale data. Okay, so that's the point, and this is a tree, quote unquote, of all life, except you'll see these funny things, which are horizontal gene transfers. So there's not actually a truly tree-like evolution. There's things going on that are not so simple. But we still talk about trying to reconstruct the tree of life, so the underlying tree on top of which you have horizontal gene transfer happening. You really don't have to know any biology to follow the rest of the talk. Okay, but it's of interest to biologists because this evolutionary context helps you answer questions. So there's this really famous statement that says, nothing in biology makes sense except in the light of evolution. And then there's a second sentence that says, nothing in evolution makes sense except in the light of phylogeny. So we're going to go out getting phylogenies, which is a nice, clean problem. And we're going to use those to answer biology questions. But the rest of the talk is just how do we get the phylogeny. So before I go on, how many of you are coming from the biological side? Almost everyone. Okay. So the great thing is that you'll actually understand the questions. And this thing is that you don't have to know any biology to follow this. Okay, that's true, but at least you can appreciate the answers. So I love talking with biologists because you understand the point. But please do ask questions. I'll try to get through everything really quickly, but if I have a clarifying question, just ask. Okay, so phylogeny estimation is a statistical problem. There's lots of big data issues. So data are, in this case, genome data, but you might also have morphology. They're heterogeneous types, and they actually have heterogeneous evolutionary processes operating in them. The large data sets, they're noisy. Sometimes your input is like from sequencing technologies where you don't actually really know what the nucleotide is. There's all sorts of stuff going on. There's streaming data. There's lots of model misspecification. So you're doing statistical estimation, but you have model misspecification. The approaches that you take from the computer science side or the statistics side, everything's NP-hard. So you're trying to solve NP-hard problems, and of course you can't really solve an NP-hard problem exactly. So you're going to end up dealing with heuristics that are going to try to search through an exponentially sized space. These are hard problems. You need to bring in all sorts of stuff, and there's lots of interesting kinds of mathematics that come in. Probabilistic analysis of algorithms, graph algorithms, all sorts of stuff. So from a computer science, math, stats side, it's extremely interesting stuff. Okay, so this talk, I'm going to give you a quick introduction to what statistical estimation is in this context. And then I'm going to show you specifically what we're doing for species tree estimation to make it more accurate. The focus here is on accuracy. How do we get highly accurate results on really large data sets? We're not focusing on how do we speed up existing methods. We're talking about how do we get highly accurate results on large data sets. To some extent, we do that by trying to take the best methods and scale them. But we're really focused on accuracy, okay? And then I'm going to be talking about some techniques hidden in blue. Astral is a method for getting species trees, and NJ-merge and tree merge are methods for scaling methods to large data sets. So the work on NJ-merge and tree merge is joint with my grad student who's here. So Erin over there, she can answer questions about the stuff that relates to what she's been doing. Okay, so here's the pipeline. At the top level, of course, first you start by saying, what are the data that you want to gather? But then you decide, I'm going to get these species, I'm going to get these genes, and you go out and you gather your data. So that's your first start. Then you are assembling and annotating genomes, and then you're computing multiple sequence alignments for each of the genes that you want to look at. And then you are computing what do we call gene trees, which is trees on the individual genes. And then you're computing a species tree from what you've done before. And then you're doing some post-tree analyses where you answer all the biology. And what I'm mainly going to talk about today is how to get the species tree. But to understand how to get the species tree, you really have to understand something about gene trees. So I'm going to start with gene trees, then talk about species trees, and then talk about how we make them better. Okay? So, by the way, the talk is actually posted on my webpage, so you can just go ahead and download it. Okay, so gene trees and species trees. So for all the biologists in the audience, you actually probably already know this, but if you open up like a molecular evolution textbook, you see the canonical thing like Jukes Canner. How many people know Jukes Canner? Everyone. Okay, so Jukes Canner evolution, it's sequences evolving with speciation and substitutions. And so you're imagining you're taking the genomes at the root is like the common ancestor of all your organisms. You're just tracing how that genome is evolving via speciation and substitutions. And you're seeing DNA sequences at the leaves, which are just from your organisms. One thing you're going to notice is that this cartoon, the sequence doesn't get any longer or any shorter, right? It's just always the same length because the only thing that's happening are substitutions. Nucleotides are changing into other nucleotides. And each of the positions is evolving under exactly the same statistical process. Okay? So that's the assumption that's underlying all phylogeny estimation, is that the sites are evolving under exactly the same model. Now, some might be faster, some might be slower, but they're really just under exactly the same statistical model. So because you're making these assumptions, you can do something like take the sequences that you see at the leaves and try to infer the tree. Now, the funny thing here is that this tree is not rooted, right? But the tree that generates the data is rooted. So the reason that you're getting an unrooted tree is that the statistical models are time reversible. You can't locate the root. So you're considered to be an accurate estimation if it matches the underlying tree, except for the location of the root. Okay? I hope everyone has sort of got that. We're trying to get the unrooted tree, the unrooted version of the true tree that generates the data. So it's a statistical inverse problem. Okay. So to make this sensible, we have to have a statistical model. So this is like the standard statistical model is that the sites are evolving under exactly the same process, perhaps with rates. And the simplest of these models is the Jukes-Canner model. The detail here is not important. The important thing is that this is a simple model. You can make more complex models. But they're all assuming that the sites are evolving under exactly the same process. Okay? Phylogeny estimation, take the sequences, infer the tree. There are many methods to infer trees from sequence data under these models. And we can ask about error. So we could do a simulation. And if you do a simulation, you have the true tree. It generates data. The data are then used to infer a tree. Then you compare the true tree and the estimated tree. So the important thing to see here is that you can quantify each. You can say is an edge correct or not. So this edge splits S1 and S2 from S3, 4, 5. The same bipartition appears in the true tree. So that's a true positive. This edge is S3 and S5 from 1, 2, and 4. It doesn't exist in the true tree, so it's a false positive. Similarly, that's a false negative. And you can quantify two types of error, false negatives, false positives. And when both trees are fully resolved, otherwise known as binary, then it's the same number of each. And the only edges that matter are the internal edges. So this has one edge right, one edge wrong, 50% error. You'd like 0% error, which means you get the right tree, right? So the error rate we're going to report is the fraction of the edges that we fail to recover. Okay? So statistical consistency is a statement that as you have a model tree, you're generating data, and as the data goes to infinity, which means to say the sequence length is going to infinity. The number of leaves is constant. The sequence length is going to infinity. For each sequence length, you can try to estimate the tree. Now, if you quantify the error, remember it's the fraction of the tree you fail to get, if that error goes to 0, that's good. If you can prove that for every model tree, the error will go to 0 as data goes to infinity, that's a mathematical proof. It's not a simulation. If you can prove it, then you have a proof that the method is statistically consistent. Statisticians like statistically consistent methods. Okay, so we can ask things like, is the model tree identifiable, which means is it actually recoverable from its probability distribution? Yes or no? The model, it's a question about the model. Some models are, some models aren't. Jukes Cantor is, general time reversible is, not all models are. But now, if a model is identifiable, you can say, is your method statistically consistent? Some methods are, some methods aren't. How much data does the method need to recover the tree correctly with high probability? That's a data requirement, otherwise known as sample complexity. Some methods are consistent, but they need an enormous amount of data. Some methods need less. Okay? This is all about statistical estimation. The last question is running time, right, and memory. So that's like a different kind of question. So what is it that we know now? We know a lot about the statistical stuff. We know which models are identifiable. We know which methods are consistent. We know a little bit about data requirements. Okay? These are all things that you need to look at mathematically. But the take-home message is that the statistical models that are identifiable are really limited in heterogeneity. They're really simple. And for those of you who want to have a conversation about that later, we can talk about it. But all of the statistical models that are used that are identifiable are simplistic. Paul, hey. Sorry, just saw a friend. And the heterogeneity is limited. Okay? So that, yes?
Could you give us a feel for the size of the problem? How many sequences?
Okay, so the tree of life is expected to have, I don't know, 8 million species. And you're trying to solve NP-hard problems, and it gets worse.
But, and the data on the...
It's just genome scale, that's it. You may think that's a lot, but it's not a lot. Okay, so computational issues, maximum likelihood, NP-hard problem, Bayesian estimation even harder because you have to let it converge. So you really have these problems about running time. And then parallelism can help, but it really doesn't address these issues enough. So what I'm going to show you is some ways of addressing this that can use parallelism, but can go beyond just what you get by parallelizing an existing method. Okay, so genome scale data, back to statistical consistency and to your question, Mike. So, you know, statistically consistent methods are guaranteed to recover the tree if you have enough data. So the hope has been to take genome scale data, and that would be enough. And I'm going to show you it's not enough, but we want genome scale data because maybe that'll help us get the true tree. So instead of having just one gene, you concatenate all 15,000 genes you have, okay? And then maybe you can get the true tree from all that data. Okay, phylogenomics is built on that assumption that you could do it. The problem is gene tree discordance, and this is where I'm saying it gets worse. So the different parts of the genome have different trees. And when I say they have different trees, it's not that you made a mistake in your estimation. It's that they actually have different histories. There are lots of reasons why different genes have different trees. One of them is incomplete lineage sorting, which is a population-level process. And due to time, I'm not going to be able to tell you exactly what it is. But fundamentally, you have this idea that you're tracing, let's say, alleles going back to the common ancestors. And as you trace them back, you know, you're saying which parent gave you that allele, then you go keep going back. And then in this phylogeny, I think this is Charles Darwin. I don't know what their names are. But these two individuals, when you trace them back, they are siblings in the gene tree. They're not siblings in the species tree. So the gene tree has a different topology than the species tree. And so what you have is this genome scale data. Yeah, it's a lot of data. But now you have this heterogeneity. It's not one tree on which everything is evolving. So as a statistical estimation problem, it's more complicated. So what you have is incomplete lineage sorting, which is going to happen with high probability when you have rapid radiations. And many data sets have rapid radiations. Rapid radiation means that you have speciation events happening quickly. So the plant phylogeny I worked on, massive gene tree heterogeneity that was consistent with incomplete lineage sorting. The Avian project I worked on, massive gene tree heterogeneity consistent with incomplete lineage sorting. This is just happening everywhere. Now, how much heterogeneity there is depends upon the data, but heterogeneity is present in all of these analyses. Okay, so there's a hierarchical model. You have a species tree that is generating gene trees. It's a mathematical process. The species tree is a generative model for gene trees. And then the gene trees are generating sequence data. And you're trying to infer the species tree from this mess. Now, you could just give up. You could just say, look, it's a mess, there's no tree. But in fact, it's a statistical estimation problem. There is a species tree. It's generating stuff. Can we reconstruct that species tree? So that's this hierarchical model. The species tree generates gene trees. The gene trees generate sequence data. And you would like to infer the species tree from that end process. OK? OK, so let's make the simplifying and, of course, unrealistic assumption that both things are going to infinity, the sequence length per gene and the number of genes. OK? Now, you can say that's unrealistic. I 100% agree. But under that unreasonable assumption, what can we do? OK, both things are going to infinity. We're going to try to infer the species tree from what we see at the leaves. The leaves are just sequences for the different genes. OK, the traditional approach, which is what you will see in every paper you ever read, right, that has a phylogeny, is concatenation. You take all of your data and you don't worry about heterogeneity. You just don't worry about it. You concatenate everything, you put it together, you run your favorite method. That might be neighbor joining, it might be parsimony, it might be maximum likelihood, it might be Bayesian estimation, whatever. You just don't worry about it. You get all of it into one thing, you pretend it's one tree, and you construct the tree. Concatenation. It is provably inconsistent, and worse, it can be provably converged to the wrong tree with confidence going to 100%. So you can get an analysis that, according to statistical support, you have 100% confidence in that's wrong. But in simulations, it's not so dire. In fact, in simulations, and I'll show you some, sometimes it's very good and sometimes it's not good. So it's a mixed story in simulations, but from the mathematical theory, it's bad. Okay? So there's actually a huge amount of controversy about how to do species tree estimation, with a lot of fighting going on about how to do it, okay? With some people really in favor of concatenation and others people against. And the ones who are in favor are basing it, oh, sorry about that. How do I get that to stop? How do I do this? Okay, it went away. Some people like simulations, and they trust simulations, so they're not against concatenation. Some people only want theory, so they're against it. Okay. So what are the statistically consistent methods? So to start with, you can do co-estimation. You can do like a Bayesian co-estimation that will take the sequences and co-estimate the gene trees and the species trees. That's beautiful, except it doesn't run on anything but very small data sets. You can do something called site-based methods, which are going to directly infer the species tree just from the site patterns. It has nice theory. It's not very well studied. I'll show you some results. And then there's this dominant approach right now, which is called summary methods, where what you do is you take the individual genes, and you have sequence data for each gene, and you infer a gene tree. And then you're using those gene trees as information about the species tree. Okay? That's the alternative dominant approach to concatenation. And again, concatenation is ignoring the heterogeneity, and it's not consistent. Summary methods can be consistent. Again, under the assumption that sequence length per gene is going to infinity, and the number of genes is going to infinity. Okay? So here's what about summary methods. So again, the idea is you have these gene trees, and you're going to try to infer a species tree. The gene trees here are drawn as though they're rooted. They're not actually rooted. We're going to try to infer a species tree from gene trees, unrooted gene trees, to get an unrooted species tree. Everyone with me so far? Any questions right now? OK, so I'm going to have a little bit of math. We have this hierarchical model. We want to infer the species tree from the sequence at the leaves. We're going to look at what a summary method would do. It would reverse this process. It would go from the sequences, try to estimate the gene trees, go from the gene trees, try to estimate the species trees. Now, if you had the true gene trees and enough of them, you would have a sample from the true distribution defined by the species tree. And that would let you get the species tree as data goes to infinity. But what you're actually doing is estimating gene trees and using those estimated gene trees to estimate the species tree. OK, so what's the math? I'm going to discuss this technique, most frequent gene tree. You have a species tree. It has only, 10 species in it. You have, let's say, 1,000 different gene trees. They're not all the same. And then you can just say, which one of the gene trees appears the most often? That's your estimate. Now, is that a good idea? Is the most frequent gene tree a statistically consistent estimate of the species tree? If it were, we'd be done. So the answer is it's not a good idea. And worse, you can actually be provably positively misleading. You can converge to the wrong species tree as the number of gene trees goes to infinity. So that's called the anomaly zone. Anyone who's heard of the anomaly zone, that's what it is, where the most frequent gene tree is a statistically inconsistent estimate of the species tree. You can't do that and have a reliable estimate. But you can do something else which is similar, which is just 4 species. Just 4 species, like human, chimp, gorilla, orangutan, just those 4. It's fine. So for 4 species alone, the most probable unrooted gene tree is the species tree. Okay, so you can get the correct 4-leaf species tree, unrooted 4-leaf species tree, for any 4 species. So now for the computer scientists in the audience, how do you solve 10 species? You look at every possible 4-leaf case, you figure out the species tree on every 4 leaves, and then you've got enough information to get the whole species tree by like a recursive algorithm, okay? So if you get every 4 species, you're done. And so again, if this were the 4-leaf species tree, it defines a probability distribution on gene trees, and you're just going to take the one that appears most often, and that's going to be a statistically consistent estimate. So that gives you a statistically consistent method for estimating any size species tree, okay? Quartet trees to species tree. Done. So Astral was developed a few years ago to solve species tree estimation using that insight, quartet trees. You take your gene trees, you encode them as quartet trees, and you take that encoding of quartet trees, and you just find the species tree that maximizes the number of quartet trees it agrees with. That's a statistically consistent problem, which is to say that if you solve that problem exactly, you will get the correct species tree with probability going to 1. The problem is it's NP hard. So you have a statistically consistent approach, but it's NP hard. So what Astral does is it constrains the search space. It constrains the search space using the input, and you can prove that it's statistically consistent in its technique. So it will converge to the true species tree as the number of genes goes to infinity. But 2 things, remember? The sequence length per gene has to go to infinity. The number of genes has to go to infinity. Statistically consistent. Okay, it's also highly accurate. This is a simulation study. The y-axis is error. The x-axis is the number of taxa. And we're comparing Astral in blue to MPS, maximum pseudo-likelihood, in red. And error, when they have only 10 species, they're the same. As you increase the number of species, error increases for both, but it increases much faster for MPS, right? MPS error is going up very fast. Astral error is going up slowly. 1,000 genes and up to 100 species, but you can go up to 1,000, and Astral continues to have low errors, gradually going up. So Astral has a really good non-parametric approach to species tree estimation, which allows it to be highly accurate. And what's more important or equally important is the running time. Although it goes up, it can still analyze really large data sets. This is showing analysis on 1,000 species and 1,000 genes, whereas MPS, which is maximum pseudo-likelihood, basically just can't finish in 24 hours on more than 100. So that's what we're seeing is running time advantage, accuracy advantage. Astral is very good. It's used all over the place, but it can fail on some data. There are data sets where it fails to complete in 48 hours, and the conditions where it fails have to do with heterogeneity. Okay, so Astral's great. It's used all over, but it can fail on some data sets. Okay, so this is the first thing I'm going to show you from Aaron's work. This is a simulation comparing 3 different summary methods, which is to say Astral, maximum pseudo-likelihood, and something called Astrid. Then a site-based method called SVD quartets, and then concatenation using maximum likelihood run with RAxML. So if you've ever done a phylogeny, you've probably used RAxML. This is RAxML on the concatenated alignment. When the gene tree error is low, maximum 50% error in the gene trees, the 3 summary methods have the lowest error, then concatenation, and then the site-based method. When the gene tree error is high, it's the opposite. Concatenation is the best. And the summary methods are the worst. So you have a proof of statistical consistency for some methods, but the relative performance depends upon the data. When the gene trees have high error, concatenation is actually the best method. The thing is, there's lots of actual biological data sets where you're actually in that end point, where all of the gene trees are bad, especially when you're taking genome-scale data, which gets back to data, right? Your data are very heterogeneous. When you're sampling across the entire genome, a lot of those regions are either evolving very slowly or you have a lot of alignment error or whatever it is, you're not able to get a good tree. So that's a common case. Okay, so what is the take-home message from here? The two best types of methods are either summary methods or concatenation. Right now, the site-based methods are not giving the good accuracy. So what do we know about those methods? Summary methods can be statistically consistent. When gene tree error is low, they're better than concatenation. When gene tree error is high, concatenation is better, but it's not statistically consistent. But they're actually both computationally expensive. Concatenation is worse. Okay, so this is the background. And this brings us to how do we speed it up? How do we do really large numbers of species? So this is a slide from a few years ago of a divide and conquer approach called Dactyl. Dactyl was originally proposed for doing trees without doing alignments, divide and conquer trees without alignments. You take a data set, you divide into overlapping subsets. How you divide into overlapping subsets matters, but you divide. You get trees on the subsets using your preferred method, whatever it is, and then you construct a supertree. Now, those are overlapping, so these trees are overlapping, so you can apply methods that combine overlapping trees into a supertree. And once you have a new tree, you can then iterate. You can divide into subsets using the new tree. Here's the problem. That divide and conquer approach is great when the number of species is not too big. But when the number of species is big, then you have to use supertree methods, and none of the current supertree methods run on large data sets. They just slow down, or they just don't run at all, or they give you bad results. So supertree methods, which combine overlapping trees, this is a computer science problem. You have a collection of overlapping trees. You want to find a tree that sort of maximizes the similarity to your input. Everything is NP-hard. Everything is slow, right? Okay. So here's what we did. We decided to do something where we didn't need supertree methods. And this is the thing that is the new pipeline. It's called disjoint tree mergers. So imagine that instead of dividing into overlapping subsets, you divide into disjoint subsets. Now you have trees on disjoint subsets. And because you have expensive methods, when you run them on disjoint subsets, it's not a problem, right? They're small. You can make them as small as you want. You can parallelize it. All this is fine. You get really good trees on the subsets, you want to combine them, but they're not overlapping, so you need some auxiliary information, such as a distance matrix. And this disjoint tree merger things, the first such method was NJ-merge, published in 2018. Then came INC in 2018. INC is incremental tree building, and a version of it that was implemented for maximum likelihood. This year, next week, it'll be presented. Tree merge is going to be presented at ISMB, and some others that are in progress. It's very new algorithmic divide and conquer approach. Divide into disjoint subsets, construct trees on the disjoint subsets, and merge them. Okay. Aaron's done NJ-merge, which is basically a modification of neighbor joining, and it achieves statistical consistency in these pipelines. It's polynomial time, and the only problem is it can fail. Now, when I say it can fail, it algorithmically has some challenges. So she then developed something called tree merge, which never fails, guaranteed to never fail. It's also polynomial time. It's also statistically consistent, and it's asymptotically faster inside this pipeline. Okay, so these are the two things, and I'm going to show you results with NJ-merge for the species tree thing. So here's what's going on here. We have species tree estimation with two different levels of incomplete lineage sorting, which lead to different amounts of heterogeneity. And we're trying to reconstruct the species tree. The top thing is error. The bottom thing is running time. Here you want it to be low. Here you want it to be low. What's being compared is Astral. Remember I showed you what Astral does. It's a species tree method. And Astral by itself or within NJ-merge. Okay? NJ-merge is dividing into subsets, running Astral on subsets, and then merging those trees together. At the top, with 100 taxa, 25 genes, 2 different levels of ILS, or 100 genes, or 1,000 genes, or 1,000 species and 1,000 genes. What you see is yellow versus blue, yellow versus blue. They're no different from each other at the top. So the error is not changing. The running time is where you get the advantage, especially on the large one. This is what you're getting with Astral by itself, and this is what you're getting when you put it inside the pipeline. And the other thing that's not shown here is that Astral actually fails to complete on some of these data, whereas it's not failing in this context. So you get this nice reduction in running time and no loss of accuracy. Okay, what about concatenation? It's a similar thing here, except it's even better. Because concatenation, which is not statistically consistent when you have gene tree heterogeneity, can actually have higher error. So this is an example where putting it inside the pipeline when you have high ILS, the error goes down. In this case, concatenation can't even run within the time limit. And you can always run it inside this pipeline. And look at the difference in running time. The scale here is different. Okay? The running time is dramatically different. Again, part of the running time advantage is just that you divide it into subsets, so the subsets are much faster. But the combination step is trivial. Okay, so summary. When you use this with Astral, you get no change in accuracy, but you get a reduction in running time. When you use it with concatenation, you can actually improve the accuracy, and you get a reduction in running time. So for the large-scale species tree estimation problems, you have 1,000 or 10,000 species, and you want to get a tree. These summary methods like Astral are much faster than concatenation. And when you put it inside this divide and conquer thing, you get an additional speed up. So you don't have to spend a huge amount of time. So just anecdotal. The Avian phylogeny that I worked on, 48 species, whole genomes, 14,000 genes. The concatenation analysis, anyone want to guess how long it took? 48, you know the answer, so you don't have to say. 48 species, what's the running time? How much time do we spend on 48 species? 250 CPU years. Just 48 species. But when you do summary methods, it's much, much, much faster. So from a practical viewpoint, summary methods like Astral really speed things up, and this can take it to much larger species numbers. Okay, summary. The best tree estimation methods are intensive. They're generally statistically designed, so they're doing either maximum likelihood or Bayesian. They don't work on large data sets, and tree space grows exponentially. So because tree space grows exponentially, you're going to have problems. Statistical consistency proofs are nice, but they don't actually tell you about relative performance between methods. Parallel implementations are helpful, but they're not enough to make things actually scale to large data sets. But this divide and conquer stuff can improve scalability, maintain statistical consistency, and can maintain accuracy, or in some cases a little bit, but not much. And finally, that divide and conquer is highly parallelizable. So all of this can be game-changing for large-scale systematics. Okay, future work. Phylogeny estimation that I've shown you right now is just talking about incongruity between gene trees because of incomplete lineage sorting. That's the least of it. There's gene duplication and loss, there's horizontal gene transfer, there's hybridization. There's all sorts of stuff going on. And if you actually want to incorporate real heterogeneity across the genome, it gets more complicated than I've shown so far. Another thing is sequence analysis from third-generation sequencing technologies. You get these really short reads. How do you classify what that read comes from? You get a read from a microbiome sample and you want to know what species it is. It may be something that you don't have in any database. Maybe it's a completely new species. How do you tell? You have to be able to deal with phylogeny estimation, multiple sequence alignment, with data that have errors. So again, this data stuff is always the key thing. It's not just statistical estimation, it's the nature of the data. And high-performance computing can make a big difference here. So this was work largely with two students, Siavash Mirarab, who did Astral, who's now at UCSD, and Aaron Molloy, who is still with me at Illinois for a few more months, and that's about it. The papers, every paper is on my website. Most of them are downloadable, and the presentations, including this one, at the website. And thank you very much. So I sped through it, and I apologize, but maybe you have questions that you want to ask now. Yeah.
Can you elaborate a bit about what you used to join the data sets? You said something like similarity matrices, but what exactly?
Would you repeat the question?
So when you have disjoint,
Non-overlapping.
right? You have disjoint, non-overlapping. What do you use in order to put them together?
Okay, so how does NJ merge operate? I'll start with that. So you have these disjoint trees, okay? But you also compute a distance matrix between all, you have a distance between every pair of species. And in most of these settings, you can do that. So for example, in the context of gene trees to species trees, and Astrid operates by computing a distance matrix between every pair of species, which is the average topological distance between the two species among all the gene trees. You get a distance matrix. That distance matrix is used to combine the trees. And so what's going on is that you can have trees that are disjoint, but the correct tree would blend them. And you're using the distance matrix to figure out how to put them together, including blending. So there's this method from phylogeny called neighbor joining by Saito and Ney. It's an agglomerative method that just figures out which things should be siblings. You're just modifying neighbor joining so that it obeys the constraints. So when you combine these trees, you're forcing the output to maintain those topologies. So it's a modification to neighbor joining to maintain those topologies. Okay, so it's polynomial time, and it's just using the auxiliary information of the distance matrix. So again, the idea is that you put your real effort on the statistical methods on the subsets, which are expensive, and you use something that may not be quite as accurate to merge them. But it turns out that this merger maintains a lot of the accuracy. And because the statistical methods that are expensive are only on small subsets, you can afford to run them, and they give good results. Is that enough of an answer? Okay, so it's the distance matrix that you're able to compute that's necessary. And if you can't get a good distance matrix, then you have other techniques you can use, but that distance matrix is key for NJ merge and for tree merge. Yeah?
You said the old method's like 250 CPU hours. Like, how much faster is the current method?
How much faster is?
The current method compared to the old method, you said it's like 250 CPU hours.
Okay, 250 CPU years for 48 species?
Years.
Okay, so the avian phylogeny, which had whole genomes from 48 birds, the maximum likelihood analysis with bootstrapping took 250 CPU years. At the time, the analysis we tried to do, besides that, was based upon MPS, and MPS doesn't work very well. Astral would have said, compute all the gene trees and then combine them. The combination step would be very fast. We actually haven't timed it, but my guess is that the big part of it would just be to be the gene trees. Any prediction about what 15,000 gene trees would cost with 50 species each? I mean, first of all, it's parallelizable, but even if you did it sequentially, it's not very much.
50 species, you could probably get a gene tree in an hour. With 50 species.
Not even. You could do it less than
I
an
was
hour.
thinking with bootstrapping. So
You don't need bootstrapping with Astral. So
Okay,
1 minute.
yeah.
1 minute per gene tree. Okay? 1 minute per gene tree. You get all the gene trees, then you combine them. The combination step is super fast. So basically, the framework's really changed. You can even get branch support without doing bootstrapping. So the whole thing has changed. So the running time has gone from hundreds of CPU years down to probably a week of computation.
So what's that?
And that's sequential. Sorry?
Logarithmic.
I don't know. Yeah. So these methods don't have big O, because you're using heuristics of NP-hard problems. They're just hill climbing, basically hill climbing with randomization. You can't bound the running time of the heuristic searches, but you can talk about it empirically. Yes?
So the astral thing, that one is based on just analyzing 4 trees at a time, right?
Astral, yeah. No, so you're still getting the gene trees, right? So the input to Astral is a set of gene trees. Those gene trees are encoded as their quartet trees to compute the distance to an optimization solution. But you're not computing trees 4 at a time. You're just using the fact that the gene trees are going to let you talk about quartet trees.
So you said that they're the only way to sort of even if, say, speciation is happening really fast, the only case where that really works is when you have like 4 taxa. And that's sort of why Astral like wants the ability to look at like 4 gene trees at a time. And then I wanted to ask, why is it that with Astral, it's not the case that the error goes down the more data you have, because like the error rates are like going up really fast?
No, Astral's error isn't going up. The running time was going. Okay, I know what you're referring to. You're referring to how the error is going up with the number of species.
Right, yeah.
That's not the
I think
data.
so.
When we talk about consistency, we have a fixed number of species. We have a fixed tree. What's increasing is the number of genes. So this is what's happening when you are taking this fixed number of genes but letting the number of species go up. You expect error to go up as the number of species goes up. If you fix the number of species but let the number of genes increase, then error would go down. So consistency is when the data available to you for a fixed tree, in this case a fixed number of species, is increasing. Okay? So Astral has less error than MPS, but Astral also in some conditions has less error than concatenation. And again, the choice between concatenation and Astral is really how much gene tree error is there. Right? If you have too much gene tree error, you should probably do concatenation. But the amount of gene tree error that you need before it becomes good to switch is actually fairly high. This is 50% gene tree error. This is up to 80%, and up to 80% it's worse. But when you're above 80%, it's definitely worse. And mind you, this model, I think, is even in the anomaly zone. So it's a case where concatenation is actually more accurate than summary methods, even in the anomaly zone, which is going to be shocking to people who believe that coalescent approaches are the right way. Sometimes concatenation is better. Yeah?
So I would like to know if you include multiple individuals in the estimation or just one individual per species.
So this simulation is only single individual, but Astral allows multiple individuals. For everyone in the room who, if you're doing a coalescent analysis, you often have multiple individuals. And you maintain statistical consistency using Astral, it's fine. But the simulations here are a single individual. And what Siavash Mirarab has discovered, who's the developer of Astral, is that you actually have better accuracy by sampling more genes rather than sampling more individuals. So you get a little bit of advantage with more individuals, but more advantage with more genes.
My second question is using the NJ merge or the second method that you developed. Do you in your species tree, does the error only account for the topology or
Just
also
topology.
the divergence time estimation?
We're not looking at dates.
Okay.
So the estimation here, the error is always quantified just in terms of the topology and the unrooted tree. We're not quantifying error for dates. We're not looking at the location of the root. We're not looking at branch lengths, anything. It's just the topology. So we're treating the topology as the thing you want to get first, and then you could root it, and you can infer dates, and you can do all that other stuff. But first get the tree topology.
I'm not sure I follow whether this effect of gene tree error was the case also at the end in the new method, in the conquer with non-overlapping. You showed us some results of
Right.
that.
So
And
we
some-
actually have, in the paper, we actually have these comparisons. That's right, don't we? We have it binned by gene tree error for the NJ merge and tree merge papers. For NJ merge? We don't? Okay.
But I guess what I could say on this is that in the case where because NJ merge is like a boosting technique, so you could use it to run concatenation or you could use it to run Astral, basically you should think about gene tree estimation error in the beginning, and if you think gene tree estimation error is really high, then use NJ merge or tree merge with concatenation, right? So it's,
So this
Yeah.
is not, I don't do this, so I was
Oh,
wondering.
okay, yeah.
No, so I thank you for
Yeah.
answering that question, but then how do we know where we are with gene tree error?
Okay, so the question was, how do you know how much gene tree error you have so you can choose what analysis to do? Okay, so let me give you an anecdotal from the Avian phylogeny. For those of you who know about bootstrapping, it's a way of estimating how much support you have in your input trees. So the average bootstrap support in the Avian data, the 14,000 genes, the average bootstrap support was 25%, which is pretty bad. And this is telling you that genome-scale data generally has a lot of noise and poor accuracy in your gene trees. So if it's 25% bootstrap support, the accuracy of the gene tree might only be 25%, which means the gene tree error might be 75%. And if the gene tree error is 75%, you're in this range. So you could use branch support as a proxy for accuracy, and the inverse would be error. But in fact, it's very hard to tell, because as I said earlier, you can have statistically inconsistent methods that will give you 100% support for the wrong thing. Right? So it's actually an open problem, statistical problem, to say how much error is there in your data. You just don't know. Right? And of course, there's this other thing, it's just about model misspecification. None of these things are based upon true models that fit data exactly. I just want to point out also that all of this was about statistical consistency as the sequence length per gene goes to infinity, right? What about the more realistic condition where what you have is a bounded number of sites, bounded sequence length per gene, but the number of genes goes to infinity? What happens then? Well, we answered this question about summary methods, like Astral, inconsistent. In fact, they can be positively misleading. You could say, okay, don't use summary methods, but what about concatenation? Also inconsistent. Nothing is consistent when you have bounded sequence length per gene. Now, when I say nothing, it's not true. I mean, there are some site-based methods, but the site-based methods don't give good accuracy in practice. Summary methods won't be consistent. Concatenation won't be consistent. Model misspecification is a big deal. This is an example of model misspecification. The gene trees are not accurate. So, okay, so statistical estimation in phylogeny, you could say is completely flawed because nothing is consistent. But I think that's a really negative perspective. I think that consistency is an idealized condition that is only really important if you have the true model. But even if you had the true model, consistency is something about what happens given asymptotic amounts of data. You never have asymptotic amounts of data. You always have finite data. So when it comes to statistical estimation, what you need to be able to say is, given the data that you have, and it gets at your question, can you predict what you can have confidence in? Can you give an idea about what method you should use? And given your analysis, can you really have a rigorous way of estimating what's reliable? And I think this is all open. I think statistical estimation is hard. Yeah.
What about integration? Like I know that you mentioned like challenges going forward, and that seems to me like one of the biggest ones, because like increasingly, like so I study plants, and increasingly genomic data sets are showing that, especially within clades, like within 20 million years or 10 million years, there's just like a lot of integration.
Yeah.
And so at what point does the the model of a tree sort of start to break down?
Completely. Extra slide. So here, the question really has to do here your particular question has to do with hybridization, but let's talk about horizontal gene transfer as sort of a similar kind of thing, right? You have genomic data that's being transferred from one organism into another, and so you don't have a tree. This is a simulation study that's looking at what happens when you have a species phylogeny, which is a species tree on top of which you have random HGT. And the amount of HGT can vary from none to a lot. And this is like 10 events for every lineage, Okay. As you increase the HGT, and you also have incomplete lineage sorting, you can look at estimation error. So percent error over there for different methods. Error goes up as you increase HGT, right? And at this level, error is much higher than it was down there. The worst is green, the best is red. Apologies to anyone who's colorblind. Green is concatenation. Red is Astral. So what's going on here is really interesting. Astral is a non-parametric method that's based upon quartet trees. It turns out that you can prove that the optimization problem that Astral is solving is still statistically consistent under a model that is random HDT. So what we're estimating here is the correctness of the underlying species tree, not the location of the HDT. So what this is showing is that a non-parametric method can do a good job at getting the underlying species tree. Now, if you want to do more than that and get a network, a phylogenetic network that locates where every HDT event is happening, where hybridization events are happening, that's another problem, which is very hard, right? So I'm showing here that you can still get an underlying species tree, and you can get a pretty reliable one. The second best method is in orange, and that's weighted quartet max cut. It's also quartet-based. It's just not as good a heuristic as Astral is. Anyway, so the point was, when your evolutionary process is not tree-like, what should you do? Is there any chance of getting the underlying species tree? Here it shows that you can get a pretty good underlying species tree. Which methods? Turns out the non-parametric methods are working well. Astral is non-parametric. Weighted quartet max cut is non-parametric. NGST, which is distance method, is not as good. And concatenation is not as good. But it's a good question. Any else?
Take one more question if there's one out there.
Yeah.
So if the goal is to estimate the species trees, and we have all this noise in the gene data, can you use other sources of data, like morphological data and something like that, to feed your models?
Okay, so the question is really, could you combine different types of data, like use morphology and use molecular sequence data? The answer is, I think you should. Now, how to combine it, right? Another type of data that people have talked about using and used separately is genome rearrangements, right? So you could use different kinds of events and different kinds of characteristics, and the question is, how do you combine them? My sense is that doing analyses separately, right, and then figuring out what's reliable in those separate phylogenies and then combining the final phylogenies together might help. So you don't have to take a tree that you get here as your assumed true tree. But if you can figure out which edges are reliable, you can collapse the remaining ones. And then you can use the other information to refine that. Or conversely, yeah?
At this point, the most natural point to tag this other information in seems to be the distance matrix. So you could have several distance matrices and say a mixture model of these matrices.
Except
that's like the fastest place anyway.
Except that distance methods are worse than others.
No, the distance matrix when you use to join the different trees.
So like NG merge kinds of things.
Right.
Okay, but NG merge, what NG merge is doing is separating out the taxa. We're talking about combining different kinds of characteristics of species. And so you really have to have a way of getting a good tree on a finite set of taxa without splitting it that have different properties. That's the mixture that's going on here. And I don't think NG merge would address that because it separates the taxa, not the characters. But all of these things need to be addressed. So it's a good question. Okay, I think.
Yep. With that, let's
Okay.
thank Tandy one more time for